The genetic and epigenetic alterations have got a definitive role in the development of the cancer. The cancer starts with non-lethal genetic damage. The mutation can be inherited, that occurs as germline mutations, or acquired, and the causes of these acquired mutation may be environmental, metabolic, or random causes. The damage may arise from chemicals, radiation exposure, due to viruses, or reactive oxygen species. The epigenetic alterations also contribute. For example, the oncometabolite alters the gene expression without changing the DNA sequence. There is a clonal expansion of a single precursor cell. The tumor cell arises from one mutated cell. Its mutations are passed to the daughter cells, and sequencing reveals that tumor-specific mutation such as point mutation, translocation, and copy number changes may occur. There are four major classes of cancer-related genes. These are proto-oncogenes that uh, that result in the gain of function of the cell. As a result, dominancy occurs. Then tumor suppressor gene, and the loss of this tumor suppressor gene will lead to the tumor development. Usually these are recessive, but sometimes haploinsufficiency of the gene may occur. Apoptosis regulating gene alter the death balance of the cell. There are some pro-apoptotic gene and anti-apoptotic gene. So if the anti-apoptotic genes are lost, uh, there is increased cell survival. Then there is another set of the gene, that is DNA repair gene, and the loss of this DNA repair genes, they lead to the accumulation of mutations that lead to genomic instability. So a mutated phenotype of the tumor results. The cancer is due to stepwise accumulation of the driver mutation. The cancer evolved over years through progressive mutations. Initially, the initiating mutations start the process, then additional driver mutation accumulate and create the hallmarks for the cancer trait, such as growth invasion and metastasis. There are numerous passenger mutations, but they are not directly oncogenic. The cancer stem cells also develop. These are persistent, long-term, self-renewable, and they accumulate mutations over years, sometimes decades, but uh, they accumulate for years before the clinical cancer emerges. The genomic instability that occurred due to DNA repair gene loss accelerate the rates of the mutation. Driver mutations are rare, but the passenger mutations, they are numerous. Then the additional mutations may occur. They affect the environment of the tumor. Some mutations help evade immune surveillance or reshape tumor stroma. These are not always classified as drivers, but still influence the behavior of the tumor. The non-lethal genetic damage is the initiating event of the carcinogenesis. Without causing immediate cell death, these mutations set the stage for the cancer development. So there are many sources of initial genetic damage or mutation. These may be inherited or germline mutations that are passed from parent to the children and present in all cells from birth. For example, BRCA1 and BRCA2 mutations, Then there are acquired mutation or somatic mutation that occur after birth. They are restricted to the affected cells or tissue. The mechanism of causing the acquired mutations may be environmental exposure. These environmental agents may be exogenous or endogenous. The exogenous agents are viruses such as hepatitis, such as human papilloma virus and Epstein-Barr virus. Then radiation can be another exogenous agent, such as ultraviolet radiation and ionizing radiation. Then chemical exposure such as tobacco smoke, aflatoxin, they are also related to the exogenous agents that are causing the genetic damage. Internal or endogenous agents are reactive oxygen species that are byproduct of normal cellular metabolism and that damage the DNA and protein. Then there are oncometabolites. These are the metabolic byproducts that alter the gene expression through epigenetic changes. Then there are spontaneous errors or random DNA replication errors. They are not directly linked to specific exposure and they are referred to as bad luck event. Then the epigenetic alterations are the changes in the gene expression without altering the genetic sequence, often mediated by DNA methylation, histone modification, and oncometabolites. So the non-lethal genetic damage, whether inherited, environmentally induced, or spontaneous, or epigenetically mediated, forms the first critical step in the long multi-step process of cancer development. The tumor arises from the clonal expansion. A single precursor cell acquires genetic damage and starts dividing. Initiating genetic damage occur in one cell sets the stage for the cancer development. There may be heritable mutations. The initial mutations are passed on to all daughter cell during the cell division. All tumor cells share these original mutations. Then the tumor cells population, it is actually the result of proliferation of these mutated cells. This is a mass of genetically related cells, while additional mutation may accumulate over time and the subclones of the tumor may form. The original site of the mutation remains in all cells. The detection of these mutations is by DNA sequencing methods. There may be point mutations that are recognized by single nucleotide changes. Small insertion or deletions can also be identified by DNA sequencing. Then chromosomal analysis is also used for the identification of these changes. These chromosomal analysis may reveal chromosomal translocations. The piece of chromosome break and attaches elsewhere, or the chromosomal analysis may reveal copy number changes. There is duplication of genes or deletion of the genes uh, or entire region of the gene. So a tumor begins from one genetically damaged cell, expands clonally, and carries heritable mutation that can be detected using modern genetic analysis. The four main gene classes are targeted by cancer causing mutation. One is the proto-oncogene or growth promoting genes. Their normal role is to stimulate the cell growth, division of the cell, and survival of the cell. The mutation types present are gain of function mutation, and their effect is hyperactive protein or new abnormal function. It promotes unregulated growth and survival, and its inheritance pattern is usually dominant. That means that mutation in one allele is sufficient. The tumor suppressor gene or growth inhibiting genes, they have got the normal role to inhibit cell growth, repair DNA, or trigger apoptosis. The mutation type among these genes is loss of function mutation, and effects are loss of growth control, means removing the brakes from the cell. And their inheritance pattern is usually recessive. That means that both the alleles must be inactivated to cause the cancer. Exception to this is haploinsufficiency, in which the loss of one allele reduces the protein levels enough to promote a cancer. Apoptosis regulating gene is the third class. These are the regulators of cell death, and their normal role is to control programmed cell death, that is apoptosis. And mutation type over here is gain of function in anti-apoptotic genes that will result in less cell death, and loss of function in pro-apoptotic genes that also leads to impaired apoptosis. Their effect, the cumulative effect, is the enhanced survival of abnormal cells. Then there are DNA regulators or DNA regulating or repair genes, the genome maintenance gene. Their normal role is to repair damaged DNA, and the mutation type is usually loss of function mutation. Their effect is that the DNA repair is defective due to their mutation. That leads to accumulation of mutation, and this creates a mutator phenotype marked by genomic instability. Indirectly accelerates the tumorigenesis by allowing mutation in all gene classes. So the cancer arises from a combination of mutation that activate growth promoting gene, inactivate growth inhibiting and pro-apoptotic genes, and disable DNA repair mechanism, leading to genomic instability and uncontrolled cell proliferation. This table is summarizing the four groups of the gene that are mutated in cancers. One is the proto-oncogenes. This proto-oncogene's normal function is to promote the growth of the cells and division of the cell and cell survival. The mutation type that is commonly encountered is gain of function mutation, and the effect of this mutation is excessive growth of cells or new oncogenic function. And the inheritance pattern of this group is usually dominant, and it means that one mutated allele is sufficient to produce the effect. Another group is tumor suppressor genes. These tumor suppressor genes, they inhibit the growth, repair DNA and trigger apoptosis, and the mutation type commonly encountered is loss of function mutation. The effect is loss of growth inhibition. That means the brakes on the growth are off, so the cell growth starts. Usually the inheritance pattern is recessive, and that means the both alleles are an inactivation is required for the development of this cancer. So however, haploinsufficiency is possible. Third group of the genes is apoptosis regulating genes. These group of the genes, they control programmed cell death. So as there are many types of the apoptotic gene, there is gain of function in anti-apoptotic gene or loss of function in pro-apoptotic gene, and the net result is reduced cell death and enhanced survival of the cell. The inheritance pattern in this group varies. The fourth group is DNA repair gene. Here, the function of this group of the gene is to repair the DNA damage, and the mutation that is present is loss of function mutation. And the effect is genomic instability, and that result in accumulation of mutation. So a mutator phenotype of the cancer develops, and here the inheritance pattern is recessive. This picture is summarizing the concept that carcinogenesis results from accumulation of complementary mutation in a stepwise fashion over time. Here, a single cell is present, and this single cell is starting or initiating the process of growth or division. So here this is a normal cell. When it it is or there is carcinogen induced mutation in this cell, what happens? This is initiation of the process of the carcinogenesis. So the first genetic alteration occurs, that is called initiating mutation, and this mutation may be caused by environmental carcinogens such as chemicals, radiation, viruses, or inherited germline mutation. There may be random errors during the cell division. This convert this normal cell into initiated precursor cell. So here this purple color cell is the cell that has initiated mutation, and then mutated cell. This cell has acquired some characteristics of a stem cell. It has now ability to self-renew. It is it has got the capacity for long-term survival. However, it is not yet fully cancerous. So there are some additional driver mutation that occur in this cell, and over time these mutations directly contribute to the malignant transformation. So a cancer cell is formed, and due to additional mutations that occur, there may be formation or development of subclones within the tumor cell, within the tumor substance, and there are many mutations that accumulate, and they give this tumor a heterogeneous, genetically heterogeneous characteristic. So the there is emergence of subclones, and these subclones are basically different population of cells within the same tumor, and each of these subclones are carrying unique set of the mutation. So clinically, the tumor is diagnosed at this stage. The tumor contains many subclones, and it is genetically heterogeneous. So this heterogeneity often complicates treatment and contribute to the drug resistance. And over here you can see that this occur over a long time. So this triangle is showing the time that has passed over the from the normal cells to the conversion of genetically heterogeneous cell mass. So driver mutation causing cancer progression, and passenger mutations, they cause mutation. Uh, the passenger mutation are the mutations without direct functional consequences. Both are present at this stage. So the driver mutations are the mutations that directly contribute to a cancer development, responsible for acquisition of cancer hallmark by sustaining the growth, invasion, or metastasis. There is initially an initiating mutation that occurs in a normal cell, and the first driver mutation is the initiating mutation, and it starts the transforming process present in all the tumor cells that arise from the original initiated cell alone, usually not sufficient to cause the full-blown cancer. Then there are acquisition of additional driver mutation. They require for full malignant transformation. Each additional mutation pushes the cell further towards the uncontrolled growth, survival, and invasion. Cancer develop in a stepwise cumulative manner, and timing of these mutation accumulation may be uh, even decades. For example, in childhood acute lymphoblastic leukemia, initiating mutation may exist long before the clinical disease appear. Even certain aggressive cancer often have a long silent phase. The cancer stem cell is the initiated cell. Often, uh, this initiated cell, it often acquires stem cell-like properties. It has the tendency to self-renewal. That means it has got the ability to divide indefinitely, and it persists for a long term. So long-term persistent and self-renewability, that are the characteristics of stem cell, are now present in the cancer cell that has got the initiating mutation. This is the cancer stem cell, and these cells serve as a reservoir for ongoing mutation accumulation over time. So a silent period before the clinical diagnosis is present. Cell with driver mutation exist before, but do not they form a detectable cancer. This latent phase is this latent phase, it lasts for years. So the cancer development occur through a gradual accumulation of driver mutation in long-lived stem cell-like precursor, often over many years, eventually giving rise to clinically detectable genetically complex tumors. Now the accumulation of the mutations in the tumor and the concept of the genomic instability. There is loss of function or mutation in the gene that maintain the integrity of the genome. So the loss of function mutation in DNA repair gene, these DNA repair genes are actually, they are responsible for maintenance of the genomic integrity, and the loss of function mutation in this genes, they impair the ability to repair the DNA damage, allow mutation to accumulate unchecked over time, and are often an early event in carcinogenesis, especially in the solid tumors. So this leads to the genomic instability, and the loss of the genomic stability leads to increased overall mutation rate, accelerated accumulation of both the driver mutation that directly promote cancer development and passenger mutation that have got no direct functional consequence. So driver mutations are relatively few in number. They actively contribute to the malignant transformation and affect cellular pathway that result in proliferation of the cell, survival of the cell, apoptosis, decreased apoptosis, and impaired DNA repair and immune evasion. While the passenger mutations, they are much more numerous than the driver mutation. They do not contribute directly to the cancer behavior, and they represent random bystander mutations accumulated over due to defective DNA repair. Then the final tumor burden is determined by the hundreds to the thousands of the passenger mutation that have accumulated and only a small number of the true driver mutation. The loss of genomic integrity accelerates both the driver and passenger mutation, resulting in tumor with a high mutation burden but relatively few mutations that actively drive cancer progression. In addition to the driver mutations and passenger mutations, there are other mutations present in the or involved in the tumorigenesis, and these mutations are primarily they affect the environment and external interaction of the tumor. We know that these driver and passenger mutations, both of these driver and passenger mutations, they are involved in the primarily involved in the cellular internal behavior such as growth, death and division, but these other mutations that are present, they are they affect the tumor interaction with the environment. So they are responsible basically for external interaction of the tumor, immune invasion, tumor stroma interaction and other mechanisms. Mutation that help tumor, they avoid immune detection, they suppress immune responsiveness, for example via immune checkpoint pathways, and they resist immune mediated killing. Then tumor stroma interaction mutation may alter how tumor cells interact with the stroma, that is supportive connective tissue, fibroblast, endothelial cells and extracellular matrix. These changes can enhance tumor survival, invasion and angiogenesis. Other mechanisms include alteration that affect the inflammation, angiogenesis or metabolism of the environment, indirectly support the tumor progression. These mutations are not classified as driver or passenger mutation because they don't directly alter the intrinsic behavior of the cell. Instead, they modulate the tumor microenvironment or host response. So, not all cancer related mutations fall into the driver or passenger categories. Some act by reshaping the tumor microenvironment or evading host defenses, contributing significantly to tumor progression. So, evolution of the tumor and progression is under the Darwinian selection theory, that is the survival of the fittest. In the initial clonality, the tumor begins from a single transformed cell, and early in development, all tumor cells are genetically identical. So they are making a clone. So the tumor origin is clonal. Then the tumor uh, growth occurs, and by the time a tumor is detectable, about size, about the weight of the 1 gram mass, or at least 30 cell proliferations or cell doublings, or 1 billion count of this cell should have should have been attained. So this is the underestimate about 1 billion, because most of the cells, or some of the cells, they may undergo the process of apoptosis during the early stages. So there are then acquisition of other mutations, and as the tumor growth, there is acquisition of new mutations, and the early mutator phenotype is now have got many mutations, many additional mutations due to defective DNA repair genes or etc. So a genetically heterogeneous tumor is formed. They are genetically heterogeneous, contain many different subclones, and each with unique genetic profile. So subclones of the tumor, they compete for the nutrients, oxygen, space in the tumor microenvironment, and therefore the most fit subclones survive and proliferate. This natural selection is the form of Darwinian evolution within the tumor. So the tumor become more aggressive over time. The emergence of dominance, more dangerous subclones are formed, and this process is known as tumor progression. So finally, we can summarize that though the tumors arise from a single cell, they evolve genetically through repeated mutation and selection, leading to heterogeneous subclones that compete and progress toward more aggressive forms. So this concept can be rephrased as during the initial clonality, the tumor cell start from a single transformed cells, and these are genetically identical. Then the tumor growth starts, and tumor growth will be detectable clinically after the cell number is progressed to about 1 billion or 30 cell doubling. And there are many cells they have undergone apoptosis during the growth timeline of the tumor. Then there is accumulation of more random mutations during the growth, and the tumor attains a mutator phenotype. Then there is formation of a genetically heterogeneous tumor with multiple subclones. So the single clone is now transformed into multiple clones, that is termed as heterogeneity, that compete for nutrient and environment, and for their survival, and the fittest survives and dominate. And over the time, this clones become more aggressive and lead to progression of the tumor. So there is a skeptical question: how do we know that these subclone really exist? So this subclonal diversity was confirmed by DNA sequencing studies of multiple regions of the same tumor, primary tumor, or associated metastatic sites, especially in cancer like renal cell carcinoma. So two types of mutation identified that are the ubiquitous or founder primary mutation found in all tumor regions that are sampled, and these are present in the original transformed cells, and they represent the initiating or early driver mutation. Then the subclonal mutations or private mutation or unique mutations are there. These are found only in some tumor regions that arise from the after the initial transformation during tumor expansion and spread. These mutations help trace the lineage of the subclones. The cancer family trees are developed by using these mutations, or these mutations form species trees in evolutionary biology, and Darwin's Galapagos finches analogy is applicable to these tumor uh, evolution. So there is difference in time scale. Species divergence usually take millennia, while these tumor subclone divergence usually take about months to years, or in rapidly progressive tumors, it may be completed even within weeks. So effective treatment acts like a Darwinian filter. Sensitive clones die, but resistant subclones, they survive and they expand. Recurrent tumors are often resistant to the original therapy, and this resistance arises not because of the therapy causes mutation, but by selecting pre-existing resistant colon. So the genetic subclones within the tumor are real, they evolve rapidly and underlie both tumor progression and therapy resistance, following pattern akin to species evolution but on an accelerated time scale. So this phylogenetic tree or tumor phylogenetic tree is a schematic presentation of genetic lineage tree tracing how different mutations accumulate and diverge within a single patient cancer over time. So there is the color code used for mutation. This red one is the ubiquitous mutation found in all tumor region, represent the initiating mutation, the founding event of the transformation. Then the primary tumor shared mutation, these are this teal color, and these mutations represent multiple region of the primary tumor. Then metastatic tumor shared, and these are blue in color, mutation shared among metastatic sites. Then unique mutation are colored or coded as purple color. These are specific to individual tumor region and represent the most recent subclonal evolution. So this tree illustrate how after the initiation of the mutation, the tumor branches out genetically into multiple subclones, each acquiring distinct mutation. This structure, it mimics the evolutionary divergence. Different branches accumulate private mutation while the earlier sharing are common ones. Here in this picture, the Darwinian original tree of life sketch, the famous Charles Darwin's handwritten sketch from his notebook with the phrase "I think" at the top. the first conceptual drawing of evolution by natural selection. So, just as species diverge from a common ancestor, tumor subclones, they diverge from a founding cancer cell. This analogy emphasizes how Darwinian evolution happens not only in nature but also within tumor rapidly and adaptatively. So the summary is that tumor evolution mirrors species evolution, but both follow branching pattern driven by mutation and natural selection. Epigenetic alterations or aberrations also contribute to the malignant properties of the cancer cell. Epigenetic modifications are heritable changes in the gene expression without altering the DNA sequence. These modifications affect how genes are turned on or off. The key mechanism for these are DNA methylation, modification of histone. So addition of methyl group to cytosine base in DNA is DNA methylation, typically silences gene expression. In cancer, can lead to activation of tumor suppressor gene, while the histone modification is the chemical change, that is acetylation or methylation to histone protein, which package DNA. Depending on the type and location of the modification, these histone changes can enhance the gene expression or suppress the gene expression. The functional impact of the epigenetic change uh, can be it determine which gene are expressed, control lineage commitment and differentiation status in both normal and cancer cells. Epigenetic marks are typically inherited by daughter cells, maintaining cellular identity. The aberrant epigenetic changes in cancer can arise spontaneously like mutations, and they result in silencing of tumor suppressor gene, global disruption of the gene expression pattern, promotion of malignant traits, for example, uncontrolled growth, loss of differentiation, and the therapeutic implication of these is that these genetic mutation or these epigenetic silencing or epigenetic changes are reversible, and uh, these are targetable by DNA methylation inhibitors such as azacitidine, histone deacetylase, HDAC inhibitor, compromising path in the genetic cancer therapy. So these epigenetic alterations such as DNA methylation and histone modification, they play a critical role in cancer by altering the expression of the gene and identity of the cell. But unlike the mutation, they offer a reversible target for emerging therapies. Thank you for watching this video. If you have any query, please write in the comment box.